I'll wait for everyone to get back in the room. I see everyone in short sleeves and shorts. It must be warmer in Nanchang. Here, spring has just come. It was a very cold, dry April. But I miss the flowers in Nanchang. I like to go to the rose garden in Nanchang by the river. It's so beautiful. Okay, I think looks like everyone's in the room again. So let's continue where we were. Okay, so this was the process of hormone secretion and producing hormones. And this was the process that was specific to insulin synthesis. And remember, insulin is made of this A chain, B chain, and C chain. The A and the B form disulfide bonds that provide you with the final active insulin hormone. And this processed C peptide becomes co-secreted with the active insulin hormone, and we can measure the C peptide as an alternative readout to insulin production. So this is the B chain here on top, the A chain underneath, and then the three crucial disulfide bonds, two of them between A and B, and the third one is an intra disulfide amongst the A chain. And again, all of these disulfides are formed within the endoplasmic reticulum, an essential part of presenting and allowing proper folding of the hormone. The signal sequence gets cleaved at the endoplasmic reticulum as part of its. Yes.
Sorry to interrupt. Thank you for noise. Okay.
No, sorry, it's my fault. Sorry, excuse me. So this is the specific part for insulin synthesis. This process, and it's again made up of A, B, and C chains. We've got here's the B chain and the A chain, and then we've got these disulfides bridging the A and B chains, and this intra-disulfide of the A chain. We have the signal sequence, which gets cleaved by a signal peptidase located within the at the interface of the rough endoplasmic reticulum. And all of these allow for the correct folding and production for further processing. The final cleavage, you get cleavage of that C chain from the A and B to present a final correctly folded active insulin hormone. And these key disulfides are maintained throughout. And the cleavage event of the C peptide occurs by an endopeptidase located within the Golgi apparatus. And as I mentioned, this C peptide can then be used to monitor for insulin production. So just a little bit about what these endo and exopeptidases are. PC2 and PC3 are pro-hormone convertases, 2 and 3. That's what the PC stands for. They are endoproteases found within the secretory system. They are calcium-dependent proteases, and they recognize and cleave at lysine-arginine or arginine-arginine specific cleavage sites. So they're responsible for taking off this C chain from the A and B to separate them. Carboxypeptidase E is the exopeptidase found in the secretory granules, and it also is part of clipping off this C-terminal basic residues. So that's the final trimming event, if you will. So you get cleavage and you get trimming. A little bit about, have you ever been to a butcher? You know, a butcher presents you with meat, right? And you have to cut off the right meat, and then you cut off the fat of the meat. So you have the PC enzymes would be responsible for cutting off your piece of meat, and then the carboxypeptidase would be responsible for cutting off the fat to trim it. So you can imagine, just back to these enzymes, you can imagine that the folding of the insulin is very important to present properly the lysine-arginine or the arginine-arginines that flank the C-peptide for proper cleavage, right? You imagine if those disulfides are not formed or insulin is very long and linear, you can see that the endoproteases might not be able to recognize the sequence and get to it, right? So if I go back to just what the thing looks like, right, you can see if this isn't folded properly and these disulfides are not formed, you're not going to get access of an enzyme to the lysine or arginine. It's not going to be able to find and cut out that portion of the polypeptide appropriately, right? Especially since, interestingly, the C-peptide, the C-chain part, is located in the middle. You really need proper cleavage to get, and the formation of disulfides to get the right orientation for recognition of these peptidases for cleavage. Now, when we talk about these different, this insulin synthesis process, we talk first in terms of pre-pro, pro, and final active insulin. And just a reminder that when we talk about the pre-pro, what we're talking about is transcription of the initial insulin gene product and then subsequent processing of the full-length messenger RNA that then gives us the pre-pro, right? Then from this gene, you've got this messenger RNA which encodes this leader sequence that will later lead itself to being the signal sequence, and then obviously the peptides domains of B, C, and A. And as the pre-pro hormone is synthesized, this signal sequence or leader sequence of 24 amino acids gets recognized and is cleaved, which then gives us the full length. Now, and we talk about these in terms of pre-pro. Once the cleavage occurs, you get pro, and then once the C-peptide gets processed by those peptidases and endopeptidases, you get the full-length insulin. So the nomenclature is pre-pro, pro, and then insulin. So, what do these look like in terms of where the disulfides are forming in the primary sequence? And this is a part I just wanted to highlight, that B chain is made first, right? And A chain is made last, even though the final peptide hormone is made up only of A and B, not of C. So it's all about the folding to get the correct orientation, to get the correct disulfide formation, to create and allow folding of the final insulin. And this one, in this case, is proinsulin, right? Because we still have the C peptide attached. Again, folding has to occur to appropriately present cleavage, in this case for that PC2, PC3, and eventually the processing of the mature peptide and cleavage, trimming down, right? You get cleavage by these CPE, right? Remember, the CPE that's going to trim the fat is going to take off those last few lingering amino acids on the edge to clean up and present the final mature insulin. Now, interestingly, these peptidases are, as I mentioned to you, the pH of the endocytic system is very carefully controlled and monitored, and CPE is one of these that's sensitive to pH. So it's only active in certain regions of the endocytic system. And so cleavage only occurs when the enzyme is active, and you find insulin in the correct compartment. Then that cleavage event will release these two arginine amino acids and then give you the final product, which then is ultimately put into the secretory granules, and in response to glucose, together with proinsulin and C-peptide gets released. So let's talk just a little bit about these secretory granules. Within the secretory granules, again, remember, because these hormones are polypeptides and sensitive to cleavage and processing in the serum and the bloodstream, one of the things that helps protect them and maintain their fold is our cofactors. And one of this, in this case for insulin, can associate with the ion zinc. And this helps maintain the fold and help keep the insulin protected and active. So the secretory vesicles then contain this mature insulin, the proinsulin, and the C-peptide, and all three are released into the portal bloodstream in response to glucose stimulation of beta cells. The C-peptide is, as I mentioned to you, secreted in a 1-to-1 ratio with insulin and excreted into the urine. There's some debate about what C-peptide's function is, if it's got a biological role or not. I mean, the typical bias is typically that there is ultimately something that it's doing, because the cell is pretty efficient and doesn't usually waste making a peptide like that and secreting it. But right now it's still a bit of a debate about what C-peptide does. But importantly, it can be used by endocrinologists for monitoring insulin production. Now, remember the T3 and T4 hormones that came out of the thyroid. And remember, you had processing between the two, and because there was very different activity at the receptor. Well, the same thing is true for insulin. So proinsulin is about less than 20%, 0.05 times as potent as insulin. So less than 5% as potent as insulin at the insulin receptor. So processing of insulin is essential for getting an appropriate response and activity in response to glucose. So we've talked about the hook of the pancreas and what it looks like. We've talked about the different cells within the pancreas. We've talked about the different hormones produced by those cells. We've talked about synthesis of peptide hormones and how that synthesis is linked to trafficking, how that synthesis is linked to different organelles within the optimized peptide secreting cells. We've talked about a reminder of the cell biology behind hormone production. And we've talked specifically, obviously, about insulin and how insulin is made. And now I want to talk a little bit about hormone diversity, because hormone diversity is directly linked to this idea of the same gene making multiple polypeptides. Okay, so remember, A chain, B chain, and C chain all came from the same gene, but we made them and put it together a little bit like a children's toy with different pieces, or a sort of a puzzle. We use some puzzle pieces, but we don't use all of them to make our insulin. Well, the same thing is true in the case of creating hormone diversity within the body. The body has been very efficient about using same gene to make multiple different hormones. So this process is called gene duplication and mutation, and it's created homologous hormone families. So you can also get this hormone diversity not just by gene duplication and mutation, but also via post-transcriptional diversification by including different exons within the final messenger RNA. So you can get a single gene that can lead to two different hormones just by taking different exons and putting them together. And then you can also get hormone diversity through post-translational diversification, okay? So you can have a single gene that can give you a single prohormone, and that can give you, then lead to multiple hormones because of the processing of those prohormones, right? So imagine in the case of insulin, if you didn't have, if instead of using CPE and to take off those two arginines, you left the two arginines on, that might give you a different hormone, okay? So this is one of my favorite exam questions in comparing hormones versus steroids. So the key part of using gene transcription, duplication, and post-translational diversification can give you multiple hormones from a single gene, right? Finally, you can get different post-translational combinations, right, to give you two genes in one hormone. Now, I'm going to step through the next few slides and just give examples of each of these and demonstrate why peptide hormones are, I think, so cool and why we have so many of these different peptide hormones. So first, gene duplication and mutation. So here's an example of an ancestral gene called Vasopressin, and it's been copied and diversified to give rise to two non-peptide hormone families. And these are the Vasopressin family and the Oxytocin family, okay? So oxytocin and vasopressin genes are very, very similar, and originated from the same ancestral gene called vasopressin. Human growth hormone, or GH, which we've heard about. The same issue came from an ancestral gene and is also related to the bovine prolactin family, okay? So both of these are examples of gene duplication and mutation. So the Vasopressin the ancestral gene, can be found in lamprey and hagfish. So they're from the genus of agnathons. I'm pretty sure you find lamprey in China. I think that it's a river fish, if I remember. And I know, and you see a lot of people, right, fishing in the Nanchang, in the river that runs through Nanchang. I forget the name of it. Oh man, too long. Our hotel is right on the river, so we walk across and walk along the river all the time. So in the original Vasopressin gene, you've got this particular amino acid sequence, and here it is duplicated in across the different oxytocin family. Okay, so here's isotocin, which you find in teleus, mesotocin, which you find in birds, reptiles, and marsupials, and then oxytocin, which you find in us and other mammals. And you can clearly see you've got this cysteine, tyrosine, isoleucine repeated, right? But this fourth amino acid is varying. So in the case of the Vasopressin, the original ancestral gene is glutamine, and then or glutamate, sorry, and then for the rest of us, it's mutated in serine, glutamine, or glutamine. In the case of us, you maintain the asparagine, cysteine, and proline, but in the case of isotocin and mesotocins, this is mutated now to an isoleucine, and then you've got the glycine. Okay, so you can clearly see that small shuttle changes, point mutations, can lead to modification. And we use this in pharmacology and drug discovery to understand which amino acids are important and which amino acids are less important. And usually the ones that are conserved are the more important ones for function. We also use it as a way to identify specificity amongst the different animal types. Now, another example is here in the Vasopressin family. So again, it comes from the same ancestral gene, but this duplication and mutation is an example. So here, this you maintain the isoleucine, cysteine, tyrosine, isoleucine, mucine, but you've got mutated. The glutamate is maintained from the ancestral gene in the case of non-mammals, but the minute you get to mammals, you get now the glutamine formation. And it's interesting to compare now. Look at this mesotocin versus lysopressin, right? Very, very similar, but it only changes between the lysine and an isoleucine, right? That's the only difference between these two hormones. In the case of us, you have quite a bit of difference between vasopressin and oxytocin, where you've got now this phenylalanine instead of isoleucine, you've got now a glutamate instead of a glutamine, and you've got now an arginine instead of a leucine. So in the case of us and other mammals, you have a serious difference between vasopressin and oxytocin, even though they all originated from the same original ancestral gene. Some of you may remember, you may have been taught by Laurent Franck. Maybe you remember him, a French guy, tall French guy. Laurent, he in collaboration with a lot of actually several Chinese researchers, was studying the origin of domestication of pigs and dogs. And one of the things that Laurent is famous for is identification of these types of genes and how they change and how their function changed as dogs and pigs were moved from say, different parts of Europe or different parts of Asia for domestication, and how different breeds of, say, pigs or dogs vary historically from ancestral animals. Laurent is no longer with Queen Mary. He's moved to Germany now, but yeah, that's one of the things he was quite famous for. So some of you may remember being taught by Laurent in the molecular biology class. I'm going to turn off my video because Vuv is telling me we have a bad connection. Let me see if I can do that. No, it's not letting me turn that off. I don't know why. Let me see. No, if I stop video, I think it will. I can't share it. Never mind. Can you hear me okay? Hopefully. Okay. So, what about some of these what about oxytocin and vasopressin in terms of what do they actually look like? They're quite interesting structures visually. Just a little side note, because these are non-peptide hormones that have a sort of a what's called a lariat structure. So they're circular, which is in some ways what helps them be protective. And this is one of the reasons why I want to highlight it and highlight the importance of these disulfide formation bridges. So they've got these stabilized by these cysteine disulfide bridges. And you can clearly see that they have very, very similar structures, even though they have key changes. So here, this proline and arginine on the right in vasopressin, and here you've got a proline and leucine that are presented differently, and that's what allows them different recognition by the receptor, along with the phenylalanine and isoleucine changes. And then just visually in the ancestral gene for GH and prolactin, it's this somatosomatolactotropin gene, originally what it's called. This led to a rise of growth hormone genes, and growth hormone is quite large. It also, from the same ancestral gene, led to these chorionic somatotropin or human placental lactogens, so it became obviously somatotropins, and then eventually also you get prolactin, all again from the same gene. So how did this occur? Growth hormone genes are expressed in the interior pituitary, as we know, in these somatotrope cells. Prolactin genes also expressed in the interior pituitary in the lactotrope cells. So what we think is we got essentially different evolution between those that led to the different cell types and then the different hormones. Okay, so that's ancestral gene. Let's talk a little bit about post-transcriptional diversification that is used to present from the same gene to give us different hormones. Now here I'll use the examples of calcitonin and CGRP. So in this case, we've got a gene, in this case, calcitonin and the CGRP come from the same, what are called the calcitonin gene. You see here, you've got a non-coding exon E1, you've got coding exons C1 and C2, you've got a calcitonin encoding exon and a CGRP encoding exon, this E5, and then you've got a non-coding exon, E6 at the end. And then due to transcription, polyadenylation, and RNA splicing, all of those things that you've learned about in molecular biology, you can then lead to a messenger RNA piecing together E1, E2, E3, and E4 that gives you a calcitonin messenger RNA, right, with eventually a signal sequence and the various pieces that will get processed. So you'll get a common region, a calcitonin precursor, pre-pro hormone, calcitonin, and then a small tail at the end, and then this proteolytic cleaving again with a variety of peptidases will provide you with an amino terminal peptide, a carboxy terminal peptide, and your final mature calcitonin. Alternatively, and this occurs in the thyroid cells, remember those C cells that we talked about calcitonin, but the same gene in the brain, you get different processing. It gives you merger now. It's the same E1, E2, E3, but now we get incorporation of this E5 exon, which is the CGRP exon, along with an E6 messenger RNA poly A tail at the ends. And this gets recognized by ribosomes in a way to make eventually the precursor to CGRP. They have the same common regions, so you get some of the same enzymes were involved for processing, but this leads to a mature CGRP in this case, and not a mature calcitonin. This is the case for the brain. And then this CGRP, there's a lot of interest in CGRP for the treatment of migraines. And with several drugs recently approved in the US for CGRP and some clinical trials ongoing globally. So, how does this post-transcriptional diversification occur so that we can appreciate all the intricacies and remind ourselves in the importance of the molecular biology that you learned earlier? So again, we've got different exons all together. We've got a unique poly A site. By cleavage and polyadenylation in the thyroid, we get this 1, 2, and 3 exons put together with our poly A, and in the brain you've got 1, 2, 3, 5, 6 in poly A. The spliceosome, which you may remember, which is responsible for piecing together different transcription fragments to make final mature messenger RNA, and the spliceosome builds together these nice messenger RNAs for us to then provide translation of calcitonin. So the mature transcripts for if you were to perform, you remember some of you did your final year projects using RT-PCR, right? If you wanted to tell the difference between the messenger RNA of calcitonin and CGRP, what region would you pick your primers for, right? If you picked your primers in the region of 1 and 3, 1, 2, 3 exons, you would get the same readout, right? So to tell the difference between the two, you would have to design your primers for CGRP in this region 6 or 5, and then for calcitonin, you would have to design your primers somewhere in this poly A for exon 4, right? So what about post-translational diversification? So I talked there about how the spliceosome performs its role in different piecing together like a puzzle, different exons. What about the post-translational side of things? Can we take advantage of post-translational modifications to create hormone diversity and create different hormones? So in this case, this gene is called the POMC gene, or POMC. Its formal name is pro-opiomelanocortin. And it's then processed into a nice messenger RNA and a giant polypeptide. And importantly, this polypeptide then gets cleaved in a multitude of different ways to give us all kinds of different hormones. Look at all these hormones we can get from a single polypeptide. So we get what's called gamma MSH from this 1 and 2. And these are some of the receptors my lab also works on. You can get ACTH for your adrenal activation. You can get beta lipotropin. Beta lipotropin can get broken up, and these are products in the corticotrophs. Okay, beta lipotropin also can get produced or cleaved within different products in the brain and some of the POMC neurons for gamma lipotropin and beta endorphin. And then gamma lipotropin can ultimately lead to the production of beta MSH and MET and caphalin, which are also ligands for some of the for other GPCRs. And then ACTH can also then get cleaved to produce alpha MSH and this CLIP peptide. So this is an example where all the variety and diversification occurs via at the post-translational level, okay? Now, where do these occur? So I mentioned some of them, but most importantly, you see this happening, this diversification happening in differently from the pituitary, also some of these in the hypothalamus, and then in some in the non-pituitary tissues, one of these being the hypothalamus that I mentioned to you. But you also can see it in other cell types as well. Skin cells also produce alpha MSH. So what, importantly, not only do you get post-translational modifications, but you also get post-translational combinations. So some hormones are heterodimeric and formed by the combination of two distinct gene products. So it's different than insulin. Insulin, remember, we had A, B, and C that all came from the same gene, and you got cleavage and you got disulfide formation between A and B chains. But imagine if two different genes formed A and B. Well, that's the case for things like follicle stimulating hormone, luteinizing hormone, human chorionic gonadotropin hormone, thyroid stimulating hormone, and all other heterodimeric glycoproteins that have typically an alpha subunit with a specific beta subunit. Those are labeled in that way, so you know that they're actually different gene products. And what does that look like? And this allows you to mix and match different genes. So imagine you've got a gene that's making a beta subunit of TSH, and then a beta subunit of FSH, or a beta subunit of LH, and a beta of HCG, and they all use the same alpha subunit, okay? And by mixing and matching different pieces, I don't know if any of you know like a Lego, right? The Lego for the children's toys. My children love Legos. You can take a common Lego piece and piece it together with different Legos, and then this gives you different building pieces that you can put together, right? So this gives you this TSH or FSH or LH. And or this HCG. Okay, so this last this is sort of the last example I have for hormone diversification and how we can get the same genes. And just to sort of summarize what we've talked about in the 4 different types, just want to provide a summary of that, of what we've covered. We've talked about gene duplication and mutation. We've talked about post-translational diversification, okay, so the single gene and 2 hormones. That's where the spliceosome was piecing together things. We've talked about post-translational diversification, so we talked about different processing by peptidases and exopeptidases to give us from the same gene different ones. And then we've talked about mixing and matching different genes together to make one hormone. So I guess that I hope that summarizes the importance of how peptides can be so diverse and so unique compared to some of the cholesterol steroid hormones, where we don't have near as much diversity from the steroid hormones. So anyways, that's all I'll talk about today, and I think I've talked plenty. I don't want to overwhelm you, and let's take it up tomorrow, okay? And tomorrow we'll go through a little more about the physiology of some of these hormones, and then specifically back to the function of the go back to insulin and the function of the pancreas.